All right, so the next question in the physical biology exam is, what is the rate of ATP consumption per micrometer cube of cytoplasm? So this is quite a fun question. And what we're going to do here is we're going to assume that most of the energy consumption is devoted to making new protein. Okay, and how much energy do I need to make a protein? Well, imagine that you have the building blocks already, that you just need to pay the workers, right? You just need to pay energy to assemble the protein. Well, one thing that you need to remember is that there's for every tRNA that you load, you spend the equivalent of 2 ATPs. Note that this is going from ATP to AMP. And then every time you incorporate an amino acid, you use 2 more ATP equivalents to assemble the polypeptide. Okay, so what I'm telling you is that because of this, the number per amino acid of ATP is on the order of 4 ATP molecules per amino acid. Okay, so what's the number of ATP that I need for all of E. coli in this case? Well, we already know the number of proteins in E. coli. We know the number of amino acids per protein, and we just figure out the number of ATPs per amino acid, right? So if you remember, the number of proteins in E. coli is on the order of 3 times 10 to the 6 proteins. There's on the order of 300 amino acids per protein, and on the order of 4 ATP molecules per amino acid. So this brings us to, let's see, 3 times 3 is 10, so 10 to the 6, 10 to times 10 to the 8, times 4, okay? So here we are on the order of 4 times 10 to the 10 ATP molecules, okay? But this is the, once again, like as we saw in previous vignettes, this is the total amount necessary to make an E. coli, and over what period of time do you need to spend this energy? Well, so we need to figure that out. So the rate in E. coli of ATP is on the order of to do this right, 2 times 3, 9. I think I did this right. It's on the order of 4 times 10 to the 10 ATP molecules divided by let's say 1,000 seconds, right? This is a relatively fast, like a 20-minute division time. Okay, so what this takes us to is on the order of 4 times 10 to the 6 ATP molecules per second. Okay, and because E. coli, if you remember, the volume of E. coli is on the order of 1 micrometer cube, so the rate of ATP consumption is on the order of 4 times 10 to the 6 ATP per second per micrometer cube. Okay, all right, so does this make any sense? So let me show you something. Oh, this is making a mess. Sorry, I tried to be smart about where I put things, and then there you go. So this is a great this is a plot from a great article by Mike Lynch, and what you can see is that they did a survey of a bunch of different cell types that have to do with bacteria, but also unicellular eukaryotes or cells in culture. And what they asked is, what is the number of molecules of ATP necessary to build this, necessary in the cell as a function of the volume, okay? And what you see is that, sure enough, we're more or less dead on what we were with our estimate, because we estimated something on the order of 10 to the 9 ATPs per cell for a volume, so which is what's shown here, for a volume of around 1 micrometer. So what this slope tells you is that basically on average you have on the order of 10 to the 9 ATP molecules per cell per micrometer cube. And if you divide by the 1,000 seconds, you get 10 to the 6 per micrometer cube per second.